Hello, welcome to Night School. I am Aria,
And I'm Darian.
and we are so happy that you are here. We are a program of nightlife at the California Academy of Sciences in San Francisco, which is a weekly in-person event, and they mix science and culture every single Thursday night. And in other words, nightlife is happening right now at the museum. And here on Night School, we bring together scientists and artists and curators to dig into a new theme every month. And Darian, what are we talking about tonight?
Aria, I'm so glad you asked. You know how some species can be found roaming the entire Earth? Some other species have home ranges restricted to truly just a few meters across. Like, what makes the difference? Like, what makes one species trend one way as opposed to another? Well, we will find out all about that in tonight's episode because we are introducing you to a trio of organisms and people who work with them with incredibly narrow geographical ranges, but a wide array of interesting things about them.
Yes, and first up, we will hear from Dr. Adriana Hernandez, one of the Academy's own botany postdoctoral researchers. And Adriana studied an incredibly special flower for her PhD program, the Mariposa lily. And Mariposa lilies bloom all over California, but there is a species that can only be found on a specific hilltop in the Bay Area. Adriana is going to introduce us to it, as well as the other lilies that she studied over the years. And next up, we have Dr. Jenny Gumm, who is a fish biologist and facility manager with the Meadows Fish Conservation Facility. There is a fish living there that is literally found in a single water-filled limestone cave. And this fish is called the Devil's Hole pupfish. It's deeply adorable and also the star of tonight's Night School thumbnail image, so you'll see it pop up on screen a few times between presenters. And tonight, Jenny's going to share about ongoing efforts to protect the Devil's Hole pupfish and what's being done to help their critically endangered population recover. And closing out our program tonight is Ruben Tovar, who is a PhD candidate at the University of Texas at Austin. Ruben studies a group of groundwater salamanders found in extremely limited areas of central Texas. And among those is a cave dweller that's commonly known as the Texas blind salamander, which over years of evolution, you guessed it, has no eyes. And we'll hear all about those stunning salamanders and the techniques that Ruben uses to study them, and just generally about how eye loss has evolved in different salamanders through time. So really great lineup.
Yeah, sounds fascinating. As always, tonight's program is live, so whether you're learning about species with the Lilliputian distributions on YouTube, or you're on Facebook, or you're on Twitch, say hi. Say hi in the comments. Let us know where in the world you're watching from, no matter whether it's your first time joining us or you're a Night School regular. This would be episode 86 for you. We are so happy to have you here either way. We will have Q&A sessions after each speaker, so put any questions that come up in the comments or the chat. And with that, we will pass it along to Adriana.
Hi, hello. I'm really happy to be here. Thank you, Darian, for the invitation. Today, I get to talk about my first true plant love, Calochortus, the Mariposa lilies. And I'll talk about endemic species, very narrowly endemic species in California, as well as a species that I studied for my doctoral dissertation. So first, I want to start with this glorious picture of Calochortus, the genus Calochortus, commonly known as Mariposa lilies. And you can see that they vary in color and shape, in the decoration of the petals, petal spots, and hairs. Mariposa lilies are found throughout North America, in the north, going into British Columbia, east into the Dakotas, south down to Guatemala. But as you can see, they have a really strong center of diversity in California. There are about 75 species throughout, and a good 3/4 of them are found only in California. Now, Calochortus are lilies. If you picture a lily, you might recall that they're kind of vibrantly colored, and they seem to have 6 petals, but really they have 2 whorls of petal-like structures. And unlike lilies, Calochortus has a specialization or differentiation of these 2 outer whorls. So it doesn't look like it has 6 petals. It has 3 sepals is what we call them, the outer whorl, and then 3 petals is the inner whorl. And so, because they do have differentiation between the two outer whorls, the inner whorl can specialize in different ways to function, to attract pollinators, for example, or deter certain pollinators, and even to regulate climate. It's hypothesized. So generally, we categorize Mariposa lilies into 4 different floral syndromes. These are suites of traits that help us to kind of identify some of the possible ecological functions that these suites of traits play. And so the first are Mariposa lilies. These are quite large, vibrantly colored, pigmented flowers. They're kind of tulip shaped, and you might see them bobbing in the wind. Then cat's ears are smaller. They are very densely covered in what we call trichomes, or little hairs. Star tulips, which are quite the opposite. These are fairly glabrous, meaning they're lacking hairs. Also small flowers generally. And fairy lanterns are these nodding flowers that kind of form this little globe. So they're also known as globe lilies. These floral traits, again, are hypothesized to help regulate microclimate. I have a phylogenetic tree here, which essentially shows evolutionary relationships between species of Calochortus. And each branch here is colored by a floral syndrome. Generally, what we see is that each floral syndrome was derived multiple times across the evolutionary history of Calochortus, suggesting that, again, those floral traits may play some ecological role. And so one of those hypotheses is in cat's ears, which again are covered in these dense hairs or trichomes. They often inhabit cool coastal areas or cool montane areas. So these hairs might be helping to maintain heat in the flower. Star tulips have very open flowers, lacking hairs, and they almost have a horizontal positioning to their petals, suggesting that they might be, given that they inhabit these kind of moister, meadowy habitats, might be helping to evaporate some of that moisture in the flower. Now, Calochortus are geophytes, meaning they have underground storage organs. They are bulbs. So I have a long plant pictured here. This is essentially what we would see above ground. And below ground is this little bulb hiding. Now, they do reproduce asexually, cloning themselves in these little bulbs or bulbils. And this is very much an onion. Onions are bulbs as well. And so if you cut it in half, you would see some roots down here, and then a very modified flattened stem called a basal plate. This is kind of hosting where the rest of the tissues are connected. And then this fleshier part of the bulb that stores food and water are essentially modified bases of leaves. So you can imagine the rest of the leaf kind of peeking out up here. So given that these plants have underground storage organs, they can really just go dormant during the hot summers of California. And they can also wait until the timing is right for them to flower. So that might mean that a bulb stays dormant not only during the summer, but it might stay dormant for a year under a really bad rain year. And so these bulbs can kind of lend resiliency to unfavorable conditions. So that's just a quick intro on Calochortus. But today we're talking about endemism. So what is endemism? It describes the natural geographic range, typically of a species. And I do just want to point out that it does not describe the abundance or the number of individuals of a species. So just because something has a really narrow range or a narrow region in which that plant or organism is naturally located, it doesn't mean that there are few individuals in that population. But typically when we're talking about endemics, we're thinking about narrow endemics, meaning they have small, very restricted geographic ranges, such as one island, one mountaintop, or one valley, or in some of the talks we'll hear about later today, just one little pond. And so around the Bay Area, some of the species that we find with very narrow ranges are Calochortus pulchellus. This is the Mount Diablo fairy lantern, found only in the Diablo Range, pictured here. Calochortus pulchellus is found in grasslands and chaparral and scrub, oak woodland habitats as well. Some oak woodland pictured here. They're found between about 650 to 2,600 feet in elevation, which is quite a broad elevational range. This you can see as an example of a globe lily. Right? Calochortus tiburonensis also has a very narrow range just across the bay from Mount Diablo from Calochortus pulchellus. This is found on one hillside in the Ring Mountain of the Tiburon Peninsula. So it's known as the Tiburon Mariposa lily. This is a catsier. You can see that it's very densely covered in hairs or trichomes. This is found in kind of grassland serpentine habitat at about 160 to 500 feet in elevation. You can see they camouflage quite well in those grasslands. There are two Calochortus tiburonensis pictured here, and one you can see this contrast of the shadow, and the other one is hiding right here between some bordea and grass. And another super narrow endemic in southern or central California is Calochortus obispoensis, the San Luis Obispo Mariposa lily. This is another cat's ear. As you can see, the petals are very densely covered in trichomes or hairs. And it also has this striking pigmentation with golden yellow hairs and red hairs at the tips. These are found in grasslands and chaparral and scrub as well, at about 300 to 1,600 feet in elevation. So again, a little bit of a broad elevational gradient. And so why might they inhabit these super narrow ranges? Could this have something to do with life history traits, how the plants are reproducing? And so Peggy Fiedler decided to take this on by comparing 3 species with very narrow ranges to a very common plant that maybe many of you Californians have seen, the globe lily, Calochortus albus. And as you can see on this map of California, it's found all along the coast ranges and transverse ranges and in the Sierra Nevada. So she compared numerous life history traits. And what she found is that some traits didn't differ between these rare species and common species, such as the number of seeds per fruit. That was approximately the same on average. And the weight of the seeds was also approximately the same on average. And that's important to think about in terms of dispersal. But also, if you can imagine, it takes 5 or more years for many of these species to go from seed to flowers. It's important to think about these reproductive traits when we're thinking about limited ranges. Now, some of the traits that did differ between the rare species and this common species is that larger bulbs were required for reproduction, for bulbs to hit that reproductive stage. So somewhere in 3 to 5 years, seeds are going to seedlings, the bulb is growing and enlarging, and eventually that bulb may go to flower. Well, larger bulbs were required for tiburonensis and obispoensis to go to that reproductive stage. These 2 species also had fewer flowers and fruits in general, meaning they have fewer seeds, right? In general, not seeds per fruit. And they also produced fewer bulbils or bulblets, right? These clones in overall. So you can imagine the population size is smaller compared to this common species, meaning there were few chances for them to disperse. Generally, though, pulchellus, the Mount Diablo fairy lantern, was sexually rigorous compared to albus, right? So this one was producing about the same number of bulbils and bulblets and didn't necessarily have fewer flowers or fruits. And so it's still not quite known exactly what is limiting this geographic distribution of the Mount Diablo fairy lantern. But some of the traits that did differ, again, between the rare and the common, including the Mount Diablo fairy lantern, is that they required, they had poor seedling establishment. So seedlings didn't really establish themselves or, grow to become these kind of full adults. Seeds did not, germinate quite well, and they generally grew slower. And so why might that be in these 3 species? Well, all 3 of them are serpentine tolerant or serpentine endemic, meaning they have this edaphic habit, this specialization to the specific serpentine soils. Serpentine soils are magnesium rich and calcium poor, which is not common and not something that plants have generally adapted to. There are also nutrient poor soils, so low in nitrogen, potassium, and phosphorus, which are things that plants require to grow. And they're really high in metals like nickel and cobalt as well. And so there's no single adaptation that all plants exhibit, these serpentine endemic plants, to deal with these conditions. There's different kinds of adaptations that have been found in general in serpentine plants. But one example of an adaptation is known as hyperaccumulation, which is essentially the plant's ability to concentrate these metals in specific plant tissues, such as specifically roots or leaves. One example known in California is Streptanthus glandulosus, a jewel flower, which is in the mustard family, and it's known to accumulate nickel in the leaves. And so I mentioned that calochordates have bulbs. Could they be accumulating or hyperaccumulating these metals in their bulbs? Well, Peggy Fiedler decided to take on this question again and found that there was a higher level of these metals in the leaves than in the bulbs. That's interesting. So maybe it could be accumulating them in the leaves. However, she compared 5 different species, and not all 5 different species were serpentine endemics. Yet those non-serpentine endemics were also accumulating metals. So it doesn't seem to be that adaptation to serpentine endemics that adaptation to serpentine soil is specific to serpentine endemics. So it might not be an adaptation after all to accumulate these metals. Now, what are other kind of levels of endemism in Calochorus? Typically, when we talk about endemics, again, we're thinking about single species with very narrow ranges, like those I just mentioned. But when we're thinking about adaptation and generally evolutionary processes, there's utility to think about the limited distribution of other scales of biological organization beyond the level of species. That's where Calochorus venustus, or the butterfly Mariposa lily, comes in. So if you at first glance see this panel of flowers, you might guess there are several different species here. We see variation in petal color ranging from white to yellow, pink, purple, red, and just about everything in between. We also see petal spots in some individuals and others where they're lacking. And in the petal spots, we see different colors and shapes. Some are kind of red-yellow, others more red and white, others, kind of barely speckled on the petal. So Calochorus venustus is a California endemic. We see it east in the Sierra Nevada, south in the Transverse Ranges, and west up into the Coast Ranges. But California is large, so just because it's a California endemic doesn't mean it's under threat or it's rare, right? Rare. And endemism is just a way of describing the species range. So what I found as I was starting to study this species is that some populations varied in their degree of polymorphism, in their degree of variability, meaning some populations you would only find white colored individuals. And in other populations, you found individuals of just about every color right next to each other. So here's an example with reds, pinks, and whites, and here red and whites, again, just right next to each other. And so I started to wonder, what does this look like on a larger scale? Let's characterize this phenotypic diversity, or phenotype meaning what individuals look like across this distribution. And so I discretized each one of these individuals into one of 5 floral colors: white, yellow, pink, purple, and red. And what I found is that in the southern populations, you would see, again, just about every color throughout, right next to each other. But as I moved north, I found fewer and fewer pigmented individuals, both along the Coast Ranges and the Sierra Nevada, such that the northern 2 most populations are almost exclusively white. And so petal pigmentation, I also decided to plot it against elevational gradients. So the white flowers were very common throughout. In fact, if many of you have seen Calochortus venustus, you've probably seen the white one. And this has a normal distribution across elevation. So individuals in higher elevations and lower elevations, but generally many right in the middle at 1,000 meters. And at higher elevations, we start to see more pink ones and more purple ones, with the red flowered individuals being restricted to the highest, most elevations, right? So this kind of red flower is endemic to high elevations. I also found that petal spots were kind of spatially structured across longitude. So if I look at petal spots just throughout the whole range of the species, throughout California, just about the same number of individuals had a petal spot as those that did not have a petal spot. But here I've plotted this, and you can kind of read this like a map of California. So this is in the coastal or western range, the transverse range in the south, and then 3 subpopulations in the Sierra Nevada going from north, central to south. And generally what I found is the coast range individuals almost always had a petal spot, whereas the Sierra Nevada individuals almost always didn't have petal spots. So petal spots were something that are not necessarily endemic to the western coast populations, but are much more abundant in those populations. Again, suggesting there may be some sort of ecological function to that phenotype or to that pattern. And so I wondered, are these maybe different species, these different colors, or these different petal spot present absence patterns? Well, I genotyped all of these individuals, essentially sequenced each one of them, 174 of them. And if there was no gene flow between individuals, I would expect them to form clusters by color. So I would find a cluster of sequenced individuals that are red, a different cluster that's yellow, a different cluster that's white, again, all pertaining to petal color. But instead, I found that individuals clustered genetically by their geographic region. So in the south, here in the transverse ranges in purple, there's a cluster of individuals whose genes clustered together. And then 2 coast range populations, one kind of more in the south and one a little bit more north. And then 3 clusters of individuals in the Sierra Nevada as well, pertaining to southern in blue, central in the Sierra Nevada, and northern Sierra Nevada in pink. So there is gene flow between all of these individuals, despite their petal color and despite the presence and absence of their petal spots. Yet some of these phenotypes exhibit narrow endemism or limited distribution. And so could it be that there's a dispersal mechanism that's limiting the distribution of certain phenotypes or certain colors? So what I did here is to model what's called the climatic niche, or essentially, given the known distribution of each of these colors, can we predict where this individual or where these colors can inhabit, given this climate model? So essentially, if I know that something is inhabiting a very moist habitat, I'm going to look for other moist habitats throughout California and say that there's a very high likelihood that I'll find it around here. And so high probability of habitat suitability is in green, and very low probability that a suitable habitat is present is in orange or white. And so for each one of these floral colors, you can see this gradient of green, very likely to be here, or at least be suitable, rather, and orange, not very likely to be suitable, given its known distribution. And the idea with predicting dispersal and fitness here is that if dispersal is limiting the distribution of phenotypes, then I would expect a much broader range of habitat throughout California to be present than these colors actually inhabiting, right? So we see a few spots in black points here in the yellow, and a few black points here in red. But there are many other patches of green that are likely suitable habitat, yet they're not inhabiting those spaces. And so perhaps there it's dispersal that's limiting the distribution of those phenotypes. But for other colors, such as this pink and this white individuals, we see that those points where they're present are pretty much occupying the predicted suitable habitat throughout California. And so there it's a matter of fitness or habitat, just not more habitat being available for this plant to increase its distribution. And so while I might not have answers today about the significance or the ecological function of each one of these phenotypes or each one of these different color forms, I just want to highlight that these patterns of limited distribution, these patterns of endemism, are typically the first hint that there's some interesting evolutionary or ecological story behind spatial patterns. So in summary, endemism is a matter of scale. You don't hear the word endemic and freak out. It's just describing the distribution, the geographic range of a species or of a phenotype of a trait. And range is defined by numerous factors such as dispersal, life history traits, climate and soil, species interactions, and geographic barriers. So with that, I'm happy to take any questions.
Great. Thank you so much, Adriana, for that presentation. We got a lot of really thoughtful questions from our audience. Let's see, let's jump right into it. Ryan wants to know if the accumulation of metals in these plants, could that potentially make these plants undesirable to any potential predators?
Oh, that's a great question that I unfortunately don't have the answer to. Yeah, I don't know. I think it's still not well known what these plants are doing with those metals. If they're using them in some way or just kind of accumulating them in certain tissues to prevent the whole plant from kind of being intoxicated. But yeah, I think that's a great question. I'm not sure what the answer is.
Sounds like a really interesting research question. There's so much phenotypic diversity, as you said. What seemed really interesting to a bunch of our audience was the fuzziness of these flowers. Does that fuzziness confer any evolutionary advantage?
Yeah, so I mean, one hypothesis is that because a lot of those hairy species are in cooler habitats, that they might be just kind of acting as heat retention. But another thing that I found recently is that a lot of those hairs will reflect ultraviolet radiation. And that might be protecting the flower in general from UV radiation, from degradation of DNA. Another hypothesis is perhaps, a lot of Calochortus have hairs around their nectary. So all Calochortus have nectary, this sweet, yummy nectar that's calling insects to pollinate them. But sometimes insects just eat the nectar and don't pollinate them, right? And so maybe those hairs are acting as a bit of a maze to prevent insects from just kind of going straight to the nectar and spending a little bit more time increasing the chance that pollen lands on the insect.
Huh. Those are a bunch of really interesting hypotheses. It's cool that it might be like built-in sunscreen. And one final question. Mitzi wants to know, what led you to get interested in these flowers and these ranges? Was that something that happened during your PhD research, or was it a love that started before that?
Yeah, so before my PhD, I was a conservation technician at the Santa Barbara Botanic Garden. And I knew I wanted to go to grad school and I wanted to study a California native plant. But being at the garden, I got to know more and more Calochortus. And, they're beautiful, really charismatic plants. But I learned about Calochortus venustus here in the mountains and saw that there were red ones and white ones and yellow ones and thought, what's going on there? And certain people in the past had suggested that those different color forms were different subspecies. And I couldn't believe that nobody had answered that question. So I decided that I would answer that question. And it's just kind of maybe not so surprising that it's all functioning as one super variable hyper polymorphic species.
That's such a fun, like, scientist impulse. It's like, no one's asked this. I will answer this question.
There are so many interesting things in Calochortus, which I hope I showed here, and yet we know very little about them. So there's still a lot of kind of open questions that I'm hoping to get back to someday.
Great, excellent. Well, thank you so much for joining and sharing with us. Up next, we will hear from Jenny Gumm.
Okay. Am I on? Is it working?
You're on and we're good to go.
Okay. All right. Well, I'm excited to be here and tell you a little bit about some of the work that I'm currently doing. I work at the Meadows Fish Conservation Facility with the U.S. Fish and Wildlife Service, and I'm going to tell you tonight about some of the work we're doing with conservation and recovery of the Devil's Hole pupfish. I have to start by acknowledging our collaborators. This species is managed by 3 different agencies. We work very closely with the National Park Service and the Nevada Department of Wildlife. And in addition to the 3 agencies working together, we also work with lots of different universities, and we have over 25 collaborators that work on recovery and research on the species. And so everything that I'm going to tell you tonight is a collaborative effort from this group of folks over the years. So Devil's Hole is the location that we're talking about for this species in terms of its restricted range. It is located about 90 miles northwest of Las Vegas, Nevada, on the Ash Meadows National Wildlife Refuge. It is in the northern Mojave Desert, but it is actually found in Nevada, not in California. Devil's Hole itself, if you look at it from space, I guess, it really just does look like a big hole in the ground. You can actually drive by it, and the way that it's situated in sort of the foothills, you might not even notice that it's there, except for the signage. It is part of Death Valley National Park, most of which is over in California. And so you can identify it by the signage, and you can actually go visit Devil's Hole as well. It will look something like this, but you can walk on this walkway and sort of look down into the habitat. So if we get a little bit closer to Devil's Hole, it is an extreme environment in a lot of different ways. So just to orient you to the small size of this habitat, the picture on the left here, you can see a couple of our Park Service collaborators getting ready to scuba dive and count the fish in the habitat here. And then you'll see a small pool of water. Underneath this platform, which is something that we put out and every time we go out to work in the habitat, and then we remove it. So we never actually go in the habitat, we stay above it. Underneath this platform is what we call the shallow shelf area. This is an average of 36 centimeters deep at this point in time. And this is where the Devil's Hole pupfish has to do all of its fishy stuff. So all of the food resources, all of the breeding habitat is located on this shallow shelf. This is about 30 feet by maybe 10 to 12 feet. And this is why this species is known to, or sometimes said to have, one of the most restricted ranges of any vertebrate on Earth. In addition to this very small shallow shelf area, you can see another collaborator here in the water, and he is in the deep pool. So another extreme component of this habitat, it is a cave system. It was formed when a cave collapsed, and this is an opening to the aquifer. And so it's not a spring, there's no outflow. It's just a calm, quiet pool of water. And divers have been down to about 435 feet with a sight line past 500. And we have no idea how deep it actually is. In addition to just the physical components of the environment itself, there's a lot of abiotic features that also make it extreme. It is geothermally heated to 33.5 degrees Celsius. That's about 94-ish degrees Fahrenheit. There's very little dissolved oxygen, 2 to 3 milligrams per liter. It's very energy limited. In this picture on the right, you can actually see from the water surface, looking up at that platform that you can visit, but you can see the sunlight hitting the water in this beautiful blue water. This sunlight only hits the water for about 8 months out of the year, and at most for about 6 hours a day. And what that means is that for 4 months of the year, there's no photosynthesis happening. There's no primary productivity supporting the ecosystem. That means that no algae is growing, no food resources for small invertebrates, which means very limited food for the devil's hole pupfish. In addition, there are some pretty extensive and extreme disturbances that happen to the environment. Occasionally, we do get flash floods happening from monsoons that occur out here in the desert. And you can find some videos of those on YouTube from the Death Valley National Park group. We had one 2 years ago in the summer. And then there also are earthquakes. So because it's a opening to the aquifer, it's a well-connected and very large body of water. This very small pool of water actually has its own tides, like the ocean. So we have a specific prediction and tracking of the water level that goes up and down every day, and then that varies over the course of the month, just like the tides on the coast. This also means that when there are earthquakes in other parts of the world, we actually get waves in Devil's Hole. And you can find video of this online as well. There was an earthquake in the Ridgecrest, California area a few years ago in 2019 in July. And that earthquake caused 15-foot waves in this habitat. The waves were up over this rock that you can see here with the game camera on the left-hand side. So really interesting place for anything to live. And the main thing that it's known for in terms of who lives there is the Devil's Hole pupfish, which is Cyprinodon diabolus. This is one of about 130 species of pupfishes that occur in various areas. A lot of the species are endemic and live in small habitats, spring systems in the desert Southwest. The Devil's Hole pupfish is different from other species of pupfishes, though. It is smaller, so the largest documented specimen in the wild is about 42 millimeters, which is just over an inch and a half in terms of total length. They have this brilliant blue coloration that you can see in the picture on the left. And they are different in a number of other ways as well. In addition to having this blue coloration and not much differentiation between males and females, whereas most of the other species of pupfishes are very different in terms of what the males and females look like, they're different genetically. They're genetically distinct from other species that live only a mile or a mile and a half away in other systems in ash meadows. They are morphologically distinct, not only in color, but also in the picture on the right. There is usually, most fishes have another set of fins here on their ventral area. And the Devil's Hole pupfish has actually lost that set of pelvic fins. They also have bigger eyes for their head size and bigger heads for their body size. And this is called neotenous development, which also reflects some aspects of their environment. Finally, they're behaviorally different as well because of the energy limitations of the environment. Pupfishes were actually named after puppies. An early ichthyologist that was describing species was watching a bunch of territorial males being very aggressive towards each other and said, oh, they're playing like puppies, and called them pupfishes. Devil's Hole pupfish are quite a bit less active. They're a bit more chill than some of the other species, and we think that's because of the energy limitation as well. So they're different in all of these ways, which makes them interesting. And they're this tiny fish, but they're a conservation icon. They are found on Nuuvi and Nuwu ancestral lands, as I mentioned, in what we know as Nevada. They were first described by settlers in 1849, formally scientifically described in 1930. They were first protected in 1952 as part of the Death Valley National Monument, and then they were protected as part of the predecessor to the Endangered Species Act, and then the Endangered Species Act itself. Another interesting component of the Devil's Hole pupfish history is that there was a case over water rights and pumping groundwater in the area that went all the way to the Supreme Court. In 1976, there was a unanimous Supreme Court opinion ruling in favor of preservation of the water due to the fish needing it, basically, to live in Devil's Hole. So the other, another extreme component of the Devil's Hole pupfish, and due to its limitation in terms of where it lives, is the population size itself. So after the pumping was happening in sort of the late 60s, early 70s, after that was stopped, the water level did come back up, and we see the population size rising. So this top sort of bluish line is the fall population count. The lower greenish line is the spring population count. The population has been counted the same way using scuba for over 50 years now. You can see historically the fall population is quite a bit larger than the spring population, and somewhere between 400 and 500, whereas the spring is around 200. There was a pretty steep population decline in the late 90s, and then another in the early 2000s. In 2006, the population hit a low of 38 individuals, and after a bit of recovery, in 2013, it hit an all-time low of 35 individuals left in the world. So 10 years ago, there were 35 Devil's Hole pupfish left in the world. There has been some recovery in the wild population. You can see both the spring and the fall counts coming back up. Really exciting last fall. We had the first population count of over 200 in over about 20 years or so. And that was 263. Our most recent count in the spring of this year, just about 6 weeks ago, was 175 individuals in the wild population. So because of these low numbers, the threat of extinction is very high. And this study, looking at cumulative threat of extinction over the next 50 years, shows that it's predicted to go extinct within 50 years with a probability of 80 or 90%. So because of this very high threat of extinction, having a backup population in captivity has been a big aim of recovery efforts for the species. So this has been tried a few times, and this is usually what this looks like, about 1,000 gallons of water in a big concrete tank. Unfortunately, these previous attempts all failed, either to mechanical failure or in one case to a hybridization event with a closely related pupfish species. And the failure of these led to the building of the Meadows Fish Conservation Facility, which you can see here. The main feature that we have in the fish facility is on the right-hand side, and this is what we call the refuge tank. The refuge tank is where we house the captive population of Devil's Hole pupfish. The fish facility has three main aims. First, establish a self-sustaining captive population, develop captive breeding methods, and then conduct and facilitate research on Devil's Hole pupfish. I'm going to tell you a little bit about each of these if I have time, but the biggest one is this establishing a captive population of Devil's Hole pupfish. So you can see here a side-by-side view of Devil's Hole and the refuge tank, and you might notice that they look pretty similar. This approach to this refuge population was different because instead of just taking some pupfish and putting them somewhere else and hoping that all goes well, that never worked before. And so instead, the approach here is to recreate their home environment and really mimic the ecosystem as a whole in order to support their population. So you can see that it looks pretty similar. The refuge tank is only 22 feet deep. It's not 500 feet deep. We had some funding limitations there, but it is 100,000 gallons of water. Much of it's underground, and so it is a cave-like system. They actually 3D scanned the topology of the substrate substrate on the shallow shelf of Devil's Hole and carved it out of styrofoam. So it is an exact replica of the shallow shelf. And we have control over the environment in a lot of ways. So we control the temperature, the oxygen, and many other features. I think we have about 87 different things in terms of flow rates, pumps, different features that we can control for the system. We do keep the temperature a little bit cooler, and we give them a little bit more oxygen than out in the wild. So it's a Devil's Hole sort of happy or Devil's Hole vacation version. But there are some differences in the community. We have more insects than crustaceans, and we don't get disturbance events. So we actually have to go in and vacuum up the substrate when we have too much detritus starting to build up. Our workflow is that we collect eggs from Devil's Hole. Here you see Olin Feuerbacher, our aquaculturist, looking at an egg mat. You can see the egg mat in the middle here. And we put these out once a month, and we hope for the best. Devil's Hole pupfish lay a single egg at a time. A male and a female will sort of settle to the substrate and lay an egg. So we put 3 of these mats out and hope that they put some of their eggs on our mats. We never know what we're going to get. And so it's always an exciting day when we collect eggs from Devil's Hole. If we find eggs, we rear them to maturity in the lab, work on spawning methods, and then they go into the refuge tank. So here's a spawning pair of Devil's Hole pupfish, an egg, and if you can very quickly scan for an egg, see if you can find it. But in the interest of time, I'm going to show you where it is right there. We isolate these eggs, rear them individually, follow their development until they hatch, and then raise them up until they're old enough to go into the refuge tank, which is about 3 to 4 months old. This has been very successful. We've collected over 700 eggs in 10 years of efforts now, and we've produced over 300 individuals. Now, Devil's Hole pupfish only live 8 to 14 months in the wild, a little bit longer in captivity, but this means that most of these fish are an annual fish. And so we do continue to collect eggs from Devil's Hole every month in order to keep a genetic connection to the wild population, as well as to supplement the captive population. Our survivorship is very high in terms of at those early stages. So easy, get some eggs, keep them alive, put them in the refuge tank, everything should be good to go. There's quite a few other aspects of this story. Because we were mimicking the ecosystem, we have some of the predators as well. So we actually had a native predaceous diving beetle that was attacking some of the eggs, and we actively managed that. This is one of our technicians celebrating his 10,000th beetle captured from the refuge tank. And this was a really key component of getting that self-sustaining population established. So before we started trapping the beetles, we had collected only 20 eggs from the refuge tank, started that in 2018. And now we've collected probably close to 5,000 eggs from the refuge tank in the 5 years since then. Figuring out how to get these to develop and be maintained in captivity is some of our current work, developing captive breeding methods. I'm going to zoom along because I know I'm short on time, but we have established some breeding methods in a lab-like environment. And some of the key components to being able to have that be successful is understanding the environment that they need, density, types of substrate has been really important. Types of lighting has been really important. They have a different courtship behavior than many of the other species of pupfishes because they're not territorial. And so making sure that they have the right kind of environment in order to successfully court males courting females. There's different microbial communities, so being able to treat the eggs with different treatments to control microbial growth and allow the eggs to develop. And then proper nutrition. Commercially developed fish foods often have different proportions of fats and proteins and things like that that are not really appropriate for a fish that goes 4 months of the year with very little to no food availability. So we've had to develop our own foods and work with figuring out exactly what it is that they need in captivity. This has been very successful, and there's a lot of numbers here that I'm going to mostly skip over in the interest of time, but showing some of our comparative data, total number of eggs. We see a couple of refuge tank breeding events with total eggs and adults produced, and a couple of propagation room, that's our lab, events with total number of eggs and adults produced. And the thing I want to stress here is that as you see from the first to the second breeding event, we've increased our viability, our hatch rates, and our survivorship to adult and the number of adults produced. So we're learning. Every time we have fish that are breeding, we're learning how to keep them in captivity, keep them alive, keep them happy and healthy, and produce more fish, which will allow us to do more research on the species in the future. So we are doing quite a bit of research, but I want to end by just showing you the status of the refuge tank right now. Our latest population estimates are over 300 fish in the refuge tank. And we additionally have about 100 to 150 fish in the propagation room. So we currently have around 500 fish in captivity. And this is one of my favorite videos showing the refuge tank and some of our devil's hole pupfish in captivity. I'm not sure if I have time for questions, but I'd love to take them if I do.
Hey, Jenny. Obsessed with these tiny fish, and obsessed with the fact that they're basically living in a jacuzzi, but that's extreme. Yeah, definitely we have time for questions. We have tons of great ones, so I'm going to get right into it. So the big question that a lot of our audience is wondering, how did the devil's hole pupfish get there in the first place?
That's a good question. At certain times, the entire area was covered by large lakes, and as those receded, different populations of fish kind of got isolated and stuck in their habitats. We don't know how long devil's hole pupfish have been in Devil's Hole. There's a lot of different estimates, and a lot of those depend on the parameters that you put into your models. Devil's Hole itself, the cave collapsed about 60,000 years ago. And we think that the sort of age of the species is somewhere around 10 to 12,000 years, I think is the latest best estimates that we have. But there were indigenous people as well in the area, and we know that they used Devil's Hole and potentially could have been moving fish around, things like that as well. So we think they got stuck there when some larger lakes, Lake Manly over in Death Valley is pretty well known. Many of the species in Death Valley got isolated there when Lake Manly receded. And so we think something similar happened here, but there could be other ways as well.
Yeah, totally. That makes a ton of sense. And Tom is asking, do you know what caused the population decline in the late 90s? Like, was there a specific cause?
We don't know specifically. There are a lot of thoughts as to what might have played into that. We know that predaceous diving beetle that we had in the refuge tank, or we have in the refuge tank, does occur in Devil's Hole, and it had started showing up sometime in the 90s. There are also things that can happen in the environment. So when they get those earthquakes, all of the good algae and everything that is in the habitat that the fish can eat disappears. It just gets washed away into the deep. And so the timing of those events is really, really important. If you have an event like that happen right as you go into that period of time when there is almost no sunlight and algae growth to support the food web, that could contribute as well to have a big die-off. There also was human influenced things as well. I know there's a story that's well known that there were some scientists that were doing some work on the fish, and some of their traps got washed in by a flash flood, and there were some fish that perished in that event. And so it's likely a combination of a lot of these things happening all at sort of in the same time period.
Yeah,
There's now a very extensive long-term monitoring of the ecosystem, and the Park Service runs that program since about 2006 or 2009. They were starting, and so now we have a lot of data to be looking at what is predicting or influencing fish numbers. Unfortunately, that monitoring program was not in place at that time in the 90s.
Yeah, gotcha. And just generally, like how is climate change impacting this? You mentioned flash floods a couple times, like, and that timing of everything. I imagine there's a lot of things to watch out there.
Yeah, so climate change is a really big threat to the species because as the air temperatures increase, the water temperature increases. So it's geothermally heated, but it's only 36 centimeters deep. It's not deep at all. And so you do get heating of the shallow shelf. And that's on average. There are areas that it's only, a couple inches deep. And so you get areas that are quite a bit warmer on that shallow shelf than other areas. And so as the climate warms and the hot part of the summer expands, there is a limit to the temperature at which devil's hole pupfish eggs can develop and survive. And so we start to expand sort of how hot it is over the summer, and you start to decrease the recruitment period for the fish. And so you have to have enough food. We know that only happens at certain times of year, but it also has to be cool enough. And so this overlay of when it's cool enough and when there's enough food, those windows are getting smaller and smaller because of climate change.
Yeah. And in the lab at the fish conservation facility, are there things that you're able to like do to the population to help increase their resilience in the wild in any way? Like if you plan on reintroducing them?
That's a good question. We do not currently reintroduce fish into the wild, especially because the population in the wild is doing well. But it's something that we know is a tool that's used for many different fish species in particular. And so we talk about that and sort of have plans for if we were to have to do that, what that would look like. We do a lot of testing of different methods of things in the fish conservation facility. So we can test different methods to try to like cool the surface of the water. And we can do that in this backup population and things like that. We're also doing a lot of studies on sort of understanding the genetics and the genomics. Having gone through these very small bottlenecks in the wild down to 38 and 35 fish that likely decrease the genetic variation that is in the species. We know that they are incredibly inbred. We know that there's very little genetic diversity in the species. And so we're doing a lot of work to try to understand that and how that influences reproduction as well as survival. They've got some really interesting mutations that have occurred along the way in their metabolic pathways, which we do think helps them deal with sort of the oxygen level being so low and the temperature being so high and things like that. So we're doing a lot to understand the species so that we can come up with things that might help them in the long run.
Yeah, that's really amazing and sounds like absolutely amazing work. And thank you so much for sharing it all with us tonight. Really glad.
Yeah, thank you.
And finally, we're going to bring up Ruben Tovar to close out the program.
Hi, good evening, everybody, or at least it's evening here anyways, in Texas. Thank you to the hosts of Night. Sorry, here we go. Thank you to the host for the invitation. I am excited to share with y'all a little bit about the salamanders that I study. I put this kind of fun title up, Crossing to the Dark Side, and thinking about a story of caves and convergent evolution. I'm a PhD candidate in the Department of Integrated Biology at the University of Texas at Austin in David Hillis's lab. And I'd like to talk to you a little bit more about these salamanders that I study. And I think that Dr. Gumm gave a great job in doing some of the preliminary introductions by way of some of the, I guess, by way of the ecosystem that her pupfish live in. But allow me to expand on that just a little bit here. In Central Texas, we have this really unique clade of salamanders. The genus is Eurecia. And similar to the pupfish, they also inhabit karst environments. And it just so happens that they're endemic to this uplifting called the Edwards Plateau, which is composed of a carbonate rock limestone. And after millions of years of erosion via dissolution, this limestone has given way to a number of microhabitats, which we'll talk about here in a second. But just to give you an idea, here are a couple of maps from a publication by Devitt et al. In 2019. The one on your left is just kind of this broad range map outlining some of the major aquifers or contributing aquifers to the Edwards Plateau. And on the right-hand side is a list of species and some of the localities that they're found at. And so you'll notice right off the bat that they're not very wide ranging. And if you look very closely, there are in fact some of them that only have one dot in one particular area. But I think to really appreciate this, and since we're in a session on endemism and really small ranges, it's best to think about the Edwards Plateau in this kind of Z plane. And so if we can just kind of cut through the geology here and think about the Edwards Plateau in this way, there are species of salamanders from the Central Texas Eurecia clade that occupy surface habitats like Eurecia sessorum, or the Barton Springs salamander, and Eurecia nana, or the San Marcos salamander. Furthermore, though, there are species that have invaded subterranean habitats, like Eurecia raphini, or the Texas blind salamander. And what's fascinating is that there are even populations, or rather species, with populations that are polymorphic. And so you've heard that word before. Here are a couple of species that represent polymorphic populations. And on the left-hand side is Eurecia latitans, or the Cascade Cavern salamander. And on the right-hand side is Eurecia terophylla. So what's interesting, though, is that there have been a number of subterranean invasions by this group of salamanders. And here is a very quick phylogeny and some representatives of these subterranean invasions. And right off the bat, you'll notice that they seem to have similar characteristics. This is called convergent evolution. This is thought to happen when an organism or organisms occupy similar habitats and are under similar selective pressures. So what ends up happening is that they look very similar to each other and evolve these similar characteristics. And so we're going to be talking a little bit more about those characteristics here in a second. And I guess in keeping with theme of my title, I couldn't help but to think about putting together both this invasion of subterranean habitat, this kind of crazy head, alien-like looking morphology of the head of this Texas blind salamander, and just thinking to myself that, man, if you could just kind of get a good angle at this, they do kind of resemble a stormtrooper, and so it all makes sense. But what's interesting again here is that there are a couple of species, as I noted before, that are polymorphic. This is when the same species exhibits divergent characteristics. But importantly, within the Central Texas Eurecia clade, there are subclades. For example, this bottom one here in blue. The blue lines are meant to indicate the subterranean invasions of this clade. And so the subclade within within this the whole is composed completely of what we identify as obligate subterranean species. That includes the Texas blind salamander, Eurecia rathbuni, and the Austin blind salamander, Eurecia waterluensis. And so, what is my research really? My dissertation is really asking what the molecular and developmental underpinnings or what are the molecular and developmental underpinnings responsible for evolution of subterranean characteristics. Now, this is a loaded question here, and the way I like to think about this, and a number of evolutionary developmental biologists like to think about it, is this kind of hourglass, where on one end we have this amazing amount of phenotypic diversity, and so for the time being, we'll entertain this polymorphic Eurecia tridentifera. On the other end of the hourglass, there is we know of all of this wonderful genetic diversity that's associated with a number of these species. But what we don't really have a clear picture on very often is this kind of black box of development in between, where we see some amount of constraint in that organisms are often develop very similarly early in development and then diverge, creating, or rather developing, into this phenotypic diversity that we are so often drawn to as biologists. And so, as I promised, I would think a little bit about these subterranean characteristics. And for that, I'm going to bring back our star of the show here, the Texas blind salamander, Eurecia rathbuni. And when I think about subterranean characteristics, I like to just broadly categorize them into two categories. And those are either hypertrophied morphologies or characteristics and reduced morphologies and or characteristics. We can call these phenotypes as a whole. And so what is hypertrophied? Well, we can think about things that are either useful or not maybe so useful when you live and are evolving in perpetual darkness. And so here are some examples of both. Hypertrophied might include this kind of broad characteristic head of these blind salamanders. Their heads are broad. The craniofacial morphology is dorsally ventrally compressed. Their chemosensory is thought to be heightened. Their lateral line is thought to be more developed. And they clearly have these kind of elongated gracile limbs, truly alien looking. On the reduced side of things, maybe characteristics or senses that maybe we don't really need when we are evolving in perpetual darkness include the reduction of eyes, reduction in pigment because there is no sun, so you don't really maybe need any pigment to protect you from that, slower metabolism, and perhaps a reduced circadian rhythm. Again, lending itself to the fact that they don't really have a day period. And so for this talk, I'd like to focus on, and for my dissertation, I've been focusing on these two kind of alternative senses here. And so I'm going to expand a little bit on both. So we'll first start with the eye and eye reduction. And so when I initially started my research thinking about these blind salamanders and how they evolved to not have an eye, or how they developed to not have an eye, the first thing I did, as we probably all do, is go to the literature and find out what others have discovered before. And it turns out that there wasn't very much by way of eye histology of the blind salamander. And in fact, the only one of two that I found pieces of literature was published in 1900. The other was shortly after, but this one had the most detail. And here is a picture of that paper, or rather an illustration that Eigenmann drew out himself by hand, the histology of the blind salamander eye, Eurecia rathbuni. And I don't know if anybody out there has ever tried to decipher histology and tissue layers from a publication from the year 1900, but it's not super easy. All right, well, great. So I kind of had a little bit of an idea of what the blind salamander, what we're dealing with when it comes to an adult blind salamander eye. But that still left a huge void for the other salamanders, especially those that are sighted. And so for my thesis and during my master's work, I set out to describe this, and this was part of my chapter, one of my chapters during my thesis. And so here's Eurystheus sissorum, the Barton Springs salamander. And what I just want to quickly point out to y'all here is that we see on the top left picture there, we see that it has a lens. Let's see if my pointer works. Here we go. Hopefully we can see this. We see that it has a lens and it has a number of retinal layers. And so with this layer here furthest to the back being the photoreceptor layer. And so that is composed of rods and cones. And so there were a couple of other gross anatomical features that we were able to identify by doing this histology of a surface salamander, including identifying an optic lens, and optic nerve. And so this led us to conclude that, yeah, it seems like Eurystheus sissorum and Nana, which is not pictured here, both have all of the appropriate anatomical features associated with vision. So of course, next on the list was to investigate and maybe do some follow-up and augment what Eigenmann had found in 1900. And so we did. And in fact, it was pretty darn close. Some of the major things I'd like to point out about the Texas blind salamander eye, and this is in an adult, is that there does seem to be some amount of retinal layers within the eye itself. But even with that, it's completely surrounded by this black pigment epithelium, or what I guess would have become pigment epithelium. And so if there were light, it probably would be very difficult for the salamander to have any particular function within that underdeveloped and completely enclosed eye that it has. This was all published in 2021 and really set the stage for future questions. And so in pursuing these questions about gross anatomy and eye structure, I wanted to include more examples of subterranean salamanders and surface salamanders. And although histology is certainly appropriate for certain questions, it is also pretty it's relatively destructive. I mean, we are sectioning through the heads of these salamanders. I completely didn't even mention the fact that both of these species are federally endangered. And so it's not like I can go to my backyard and simply pick up one of these salamanders. So I look towards another technique that might help better understand and not be as destructive as histology. And so there's a relatively new technique that's come out, and it expands a little bit on what's already out there, known as micro-CT scanning. This particular technique that I'm using, though, is a contrast-enhanced micro-CT scanning. And what we do is we use iodine to stain the soft tissue of the salamander. And so typically during a micro-CT scan, you will receive back a scan, but for the most part, all of the dense tissue is what you see. And so that of course, doesn't answer any of my questions regarding eyes or brain structure or any other soft tissue senses that I'm interested in observing. And so to do this, it's relatively simple, and we get all of this soft tissue data back and all of the dense tissue data back as well. So you can see here, as we go through this Eurystheus rathbuni eye head, all of the muscle, all of the bone, and it's a great technique to use for something that is so relatively rare to come by. I'll also point out that these little dots all over this Eurystheus rathbuni eye head, we are going to get to that in just a moment. Okay, great. So I was able to collect a number of species Here on the left-hand side is Eurecia rathbuni, the Texas blind salamander, the blind species. On the right-hand side is Eurecia nana, the San Marcos salamander. It's considered a sighted species. And so we use this DICT approach, and in doing so, since we can see the soft tissue of interest, in this case the eye, we can then go in with software, 3D software, and segment out all of the soft tissue that we are interested in, and in this case, the eye. So here is a rendered DICT scan of each of these individuals' heads with the eyes segmented out. And so why is this significant? Because historically, what we would usually do is take a 2D photo of the head, maybe in several angles, and draw 2D lines just across maybe the eye to get some idea of eye diameter or eye width. Here, by using 3D rendering software, we're able to get we're really able to measure all of the eye and its complexity, right? So what we're able to then analyze are not 2D images, but rather volumetrics. And so we use this technique in a number of species. And so here in this plot, we have 3 salamanders over here in these 3 colors. So this is Eurecia latitans. This is, if you remember, that polymorphic population. And in this case, this is a cave population. And so this is an example of a Eurecia latitans adult head here. And then over here we have in purple Eurecia wallacei, and in green Eurecia rathbuni. And so wallacei is another new species that I've introduced you all here just right now, and that is a Georgia blind salamander. No, they do not live in Texas, but we were lucky enough to be able to collect a number of these individuals to include in this study. On the right-hand side, in blue and green here, is Eurecia sissorum, the Barton Springs salamander, and Eurecia nana, the San Marcos salamander. And so what this plot is simply showing is that the biggest amount of variation that that's being explained here is, number one, between both of the ecotypes. So all three of the species on the left-hand side are subterranean in phenotype. All 2 of the species on the right-hand side are surface in phenotype, and then we can also put in, okay, well, what is really driving this variation within this within this plot? And then what we can see is that, in fact, yeah, the lens and the retina are clearly larger in the 2 surface species than they are in the subterranean species. So that's really cool. We've been able to do that with a number of species, and we're continuing to add to this data set. And so what we've concluded from this thus far is that subterranean salamander species have this less overall eye volume. That is to say that this is an addition of both the retina and the lens, and structure relative to the surface species. So that was really nice to explicitly test that. Okay, so what about the lateral line? And the lateral line, if you remember, we're thinking might be a sensory organ that would be useful in perpetual darkness, or when you're evolving to live in perpetual darkness. So what is a lateral line? Well, the lateral line contains a series of these mechanosensory mechanoreceptors called neuromass. And so maybe some of us have seen like Shark Week or something, and so this lateral line might sound familiar. And so what we were able to do, or rather, what we were able to do is quantify these neuromass. But what do these neuromass do is that they send information to the brain about water movement around the salamander. And so they're composed of a number of cells. So here is the epithelium, otherwise the skin cell, skin layer. The neuromass has these hair cells, these mantle cells. On the hair cells themselves, they have these cilia, and particularly they're called stereocilia, and they're very similar to the same type of cells that you have in your inner ear, and are associated with your vestibular system and keeping balance. These hair cells are then encapsulated by this cupula, and whenever there's water movement or pressure that displaces that cupula and displaces those hair cells, they send a signal to the brain, telling it that they are feeling this displacement in the water. And so we can then divide the lateral line into two major regions: the anterior lateral line, which is the lateral line that is associated with the head, and the posterior lateral line, which is associated with the trunk and tail of the salamander. And so for this study, we really zoned in on the anterior lateral line. And so you can see here in this video, we are able to identify the neuromass on the surface of the skin using dye-CT scans. Then we went through and we put dots on every little bump we saw that was associated with the lateral line. And we can then, I'll replay that really quick, we can then get a visualization of the distribution of the lateral line. And we can also simply just count the number of lateral lines that are surrounding the head. And that is exactly what we did. And so we divided the neuromass around the head into 5 groups based on the earlier publication. And so this is a little bit what that might look like. We colored them differently, and those 5 groups were the orbital region, nasal region, maxillary, mandible, and postorbital. And so here's what we found. Let me show you. On the x-axis, we have some species that are different from what we've been talking about. And so there is Eurecia pallidicola. This is an important species because Eurecia pallidicola is a what we call a biphasic species. And so an important note about these Central Texas salamanders, Eurecia in particular, is that they are pedomorphic. You heard Dr. Gumm speak a little bit about neoteny, and so neoteny is a way to reach pedomorphism, and specifically, neoteny is the slower rate of somatic tissue relative to the gonad. So this is all to say that the typical amphibian life history is to have a larval stage, usually aquatic, go through metamorphosis, and then be a terrestrial animal. In our case, this group of salamanders is perpetually at this larval state while still reaching sexual maturity. However, Eurecia pallidicola is the closest relative to our group of salamanders that still goes through metamorphosis. So we included it here in our study. Here we have Eurecia nana, Cisorum. Eurecia terophylla is another polymorphic species. And so here I have indicated as the surface, this is the surface population. Latitans, in this case, the subterranean, because that's another polymorphic species. And terophylla subterranean, subterranean population, and Eurecia rathbuni, the Texas blind salamander. And so, sure enough, it seems like there's a difference between the number of neuromass, anterior neuromass, between surface and subterranean species. If we take this further, we can see in a similar plot as we saw before that all of the surface species fall on this side of the line, whereas all of the subterranean species fall on the right side of the line. Once again, indicating that 82.4% of this variation is explained simply by surface versus subterranean species, which is pretty cool. And if you're interested, here is the same rendering I showed you just a minute ago with the San Marcos salamander and all of the regional neuromass associated with it. And here is a Texas blind salamander with all of the neuromass in their respective regions and colors. And so speaking of regions, then we might be interested to know which regions might be driving this difference. And it seems like in the subterranean salamanders, they have more post-orbital neuromass. So these baby blue these baby blue dots here, and more mandibular neuromass, these yellow dots here, than the surface species do. Of course, maxillary is just behind it, and to be followed by orbital and nasal. Okay, cool. So we've tested the volumes of a number of blind species and sighted surface species, and there's a difference. We've tested the number of neuromass as an extension of the anterior lateral line system, and it turns out that subterranean salamanders have more neuromass, anterior neuromass, than surface salamanders do. That this furthermore suggests that the lateral line may be compensating, or at least be one sensory modality that might be compensating for the loss of vision in the subterranean species. Furthermore, we observe this pattern between multiple subterranean and surface species. So that is really cool. And so we're really digging into understanding how far-reaching convergence is within this group of salamanders here in Central Texas. So some ongoing work. I mentioned that I'm interested in really that missing middle piece of development. And so we are working towards continuing to build our developmental series of these salamanders to better understand how eye development is diverging between blind species or subterranean species of salamander and sighted species of salamander, and whether or not they're doing it in a similar way. And so we've been collecting a number of developmental stages to really fill in the gap to better understand this. And again, we're still using this DICT approach. And so, in trying to be as inclusive as possible with what might be, and tell the most holistic story about this fascinating group of salamanders here in Central Texas, we have been working to collect and scan soft tissue of as many species as we can fit into our data set. And I will say that one thing I left out about this particular technique is that the fixative that we use allows us both to scan these salamanders and also do downstream molecular work, which is extremely important given my question about molecular and developmental underpinnings responsible for eye loss. And so I'm excited to say that we've just gotten back each one of these species you see around this phylogeny here as an adult. We've just gotten back a good amount of transcriptomic data. So we are going to be using a expression approach to better understand which genes are being turned off and on, and which genes might be important and critical for eye development, and which genes might be important or critical for no eye development. And so with that, I'd like to thank all of these people, the Hilla Zamudio Canatella Lab at UT, the UT CT facility, which have been phenomenal in helping to really disentangle the approach of DICT scanning, the Garcia Lab at Texas State University, and NSF. And importantly, we received this NSF grant to pursue this question about this question on evolution and development as a collaborative grant between my former master's advisor at Texas State and my current PhD advisor at UT, and pulling together our resources to better understand and answer these questions. And within that grant, during the summer, we were able to dedicate a component of it for undergraduate research. And so at the bottom left-hand screen, I'd like to thank and acknowledge the NSF undergraduate summer research fellows these past couple of summers that have been instrumental in collecting a lot of this data, and I appreciate. And I cannot leave out how important it's been to have a working collaboration also with the U.S. Fish and Wildlife at San Marcos and at Uvalde. The San Marcos Aquatic Resource Center is similar to what Dr. Gumm was explaining to us earlier. Has been charged with being a refugia for a number of these endangered species of salamanders. And if it wasn't for their efforts in breeding and maintaining a lot of these endangered species, I wouldn't have the data set that I have. And we wouldn't be finding all of these really neat, interesting, and fascinating trends with the evolution of these blind salamanders. So I really have to thank them a lot for all of their help with collecting specimens. And with that, I'd love to take any questions if I have time.
Ruben, we have so many great questions from our audience, very thoughtful, all of them, and we do have time for some. So I'm thinking we can start kind of big picture questions and get a little narrower. Carrie asks, do you know how many salamander species exist on Earth?
Oh. Oh, man. I mean, not a solid number. And this is, here's also why. I don't know if you, Carrie, if you're still out there, I don't know if you noticed on the phylogeny, but a number of those, I had E-SPA, and E-SPA is indicating that it has yet to be described. So we have new species here in Texas that haven't even been described yet. And so that is such a changing number, I would imagine. So the answer is I don't know off the top of my head, but if I told you a number now, it would likely be different in like a week or so, probably.
Great. We know that a lot, as you said in your presentation, a lot of salamanders have independently evolved these kind of troglodytic characteristics, both in North America. I can think of examples in Europe and all across the world. Is there something about salamanders that makes them so well suited to adapting to cave life? Like, why do we see this pattern specifically in salamanders over and over?
Right. I would say there are studies certainly that suggest that pedomorphism is a change in life history state that has allowed for this phenotypic diversity and perhaps the occupation of these subterranean habitats and then the phenotypes that follow after that. But I wouldn't say that salamanders are maybe the only ones that do this. In fact, my inspiration for pursuing this was because I read a paper by Bill Jeffrey on Astyanax mexicanus. And so the Mexican tetra is and I didn't have time to talk about that today, but it's all part of the story. And so the Mexican tetra has been really well studied, and that is another species that has had a number of independent subterranean invasions. However, I wouldn't say that it shares the same taxonomic diversity and uniqueness as our Central Texas salamanders, but fish also seem to be able to do this. And so there are fish in China, there are fish in the Middle East, and clearly in Mexico that have invaded subterranean habitats and do have these similar phenotypes. They don't have pigmentation. They have really weird craniofacial morphology. Yeah.
Oh, it's so exciting that you're inspired by the Mexican tetra. It's a very cool species. I know we do house some of them in the Steinhardt Aquarium at the California Academy of Sciences, if anyone wants to check them out. Bethany, I know you spoke about the lateral line in great detail and how they use that as like a way to sense their surroundings. Are there any other senses in the absence of vision that they use? Like, is sound something that they're capable of sensing?
Right. So these are all on the to-do list. So with those scans, we can observe the inner ear. We can see it. I will, but before we go to the ear, my next target is the olfactory epithelium. And so the sense of smell, chemosensory sense, is likely playing a pretty significant role. So that would be my next target. And it is our next target. We have undergrads about, I think we're about to have an undergrad be dedicated to segmenting out the olfactory epithelium. So we're very interested to see what we find.
Oh, great. You spoke a lot about polymorphic species. Is there any reason that polymorphic species are defined as one species as opposed to two?
Any reason? Why they're defined as one species as opposed to two? I don't know. I think it's really in that I've always thought that the polymorphism is just defined as just one species. So I'm not sure why it is only one species as opposed to two. Yeah. That's a great question. I don't know.
Yeah, it's a totally valid answer. And just to close off the night, a lot of people took note of what Aria in our notes described as the cute little axolotl-like head thingies. Could you explain a little bit more about what those are on this salamander species?
The gills?
I believe that's what people are referring to.
Yeah, the gills. They're gills. Yeah. So I mean, they are aquatic, right? Remember, they don't leave that larval state, and so they remain in the aquifer. And that is also another reason why the aquifer, the quality of the water is so important. Just to answer that question, that it's their gills. They use it for oxygen respiration. They can also respirate through their skin, as other amphibians can as well. I think in this case, their gills are the main mode for that. Yep.
Awesome. Well,
go ahead. Sorry,
I didn't mean to cut you off.
No,
go ahead.
You're good. Oh, axolotls. Some of those populations are also pedomorphic, so that's probably why they look like axolotls.
Great. Well, we so appreciate you taking the time to shed a little bit of light on the species that lives in the deep, dark depths of the caves. Thanks. I'm going to bring back Aria. Thank you, Ruben.
You got it.
Can't believe you exposed me for not knowing what salamander gills are, but it's fine. It's fine.
It's just such a great description.
In any case, thank you so much to Ruben, Jenny, and Adriana for being here tonight. We've absolutely loved hearing from all of you and about all of your work. And thank you all out there for watching and being with us here this evening. Definitely recommend poking around to see what kind of hyperlocal animals or plants or algae or fungi live around your area. You might be surprised.
Yeah, I mean, in California, I was shocked by how many of my neighbors were like truly just my neighbors. Wild. We here at Night School are live the third Thursday of every month, which means that we will be back on July 20th to explore the myriad ways that humans have learned from and appreciated outer space, be that in the past, presently, or how we might continue to in the future. The night sky is full of exciting mysteries, whether that's comets or exoplanets or planets that are no more. So join us as we uncover those mysteries on Night School Looking Up. I looked up, I saw ceiling. It would have worked a lot better if I were outside. Aria, please make me stop talking.
Until we do bring pieces of the sky to you on this virtual platform, you can subscribe to the California Academy of Sciences YouTube channel so that you'll be notified when we come back. You can also rewatch all of our past episodes, including this one, on our YouTube channel anytime from anywhere on Earth, not outer space. Don't know about that one, but Earth for sure.
Yeah. Thank you again for joining us. See you next time. Bye.